So we're going to kick off this last day of i2eye. In the first session here, we have an invited speaker who's joined us all the way from Norway, Rigmor Baraas. So thank you for coming, and I'm going to hand over to you to moderate this session.
Thank you so much, Kate, and thank you for inviting me to come to Paris and to this exciting meeting. So I'm going to talk about between individual variation in human macular cone and RPE cell topography. I got an error message here. I don't know what to do because it's in French. Maybe I can ignore.
No, we need to share screen.
Oh, so it's not shared.
I guess it's this one.
We can see your slides.
Is it okay now?
So you can, yeah.
Yep, looks good.
Okay. You can see slides and me, and yeah.
Perfect.
Thank you, thank you. So then I'm going to start. If I can get my slides to work, and then I have to click forward like this. So I thought I had to begin with show all of you where I am from. It's in Kongsberg, which is marked on the map here. 1,290 kilometers north of Paris, not that far. And I'm from the University of Southeastern Norway, and that's a campus university with 8 campuses. And the National Center for Optics, Vision and Eye Care is at Campus Kongsberg, which is about an hour southwest of Oslo. Now I have to press. And I'm going to start with thanking all the people who I work with in Kongsberg on this area. They all work very hard, and I'm very lucky to have this team. We got funding from several sources that we're also very happy with. So the work I'm going to present is something all of these more or less have been involved in. So I'm going to talk about cone and RPE cells in healthy humans. And this is because we're very interested in understanding this transition between what's normal and healthy and what's age-related disease. So by combining imaging modalities, such as OCT and different AOSLO modalities, our aim is to make progress in understanding implications that variation might have on both eye growth and eye health. And this is of particular interest because of how the retina is shaped and how the cell topography varies from the central part of the retina and as we go out to the periphery. And what's important here is that there's a complementarity between, particularly in the macula, between cones and RPE cells. And these regional differences play out differently in health, in a healthy eye compared to a diseased eye. So let's see how I can do this now here. There's so many windows. I need to move things out of the way. And the retinal pigment epithelium, as most of you here know, is really very important for the health of the retina. But it doesn't only support the retina in a healthy eye, it also has implications in the cone cell cycle before the baby is born. And this is part of the reason why we wanted to look at this relationship between cone and RPE cells to see whether this is a relationship that's preserved into at least young adulthood. And RPE also plays a role in how the foveal avascular zone is developed. But the foveal avascular zone alone doesn't explain the shape of the fovea and the variation in the shape of the fovea between individuals. An AMD is a disease that is related to the photoreceptor support system, which also, of course, involved the RPE. In the paper that I refer to on the right of the slide here, we also suggest that maybe AMD starts with one RPE cell being damaged, and then it continues from there. But it might, of course, be a group of RPE cells that it starts with. But drusen is one of the first signs of AMD, and we have shown that drusen, even small hard drusen, affects the overlaying cone mosaic. AMD has a lot of different risk factors, and one which is related to other things is a shorter eye. But then we also know that late AMD is related to being male, being old, and smoking and diet, and also being Northern European is a higher risk. But then there's a lot of genetic risk factors associated with late AMD. More than 50% of heritability is associated with AMD, but it doesn't mean that every person who has these genes might get AMD. And how your eyes develop and how they age, it depends on many different things. Your genes, but it also might depend on where you live and who you are, what kind of things you do. But also sex and ethnicity differences are known, both in eye size, but also in human retinal structure, such as retinal thickness and foveal pit morphology. And these differences seems to be related to part of pigmentation, but more work is needed to look at this. We also know that eye size is linked with cone density. So eye size is an important factor when we want to understand these different things. So with this knowledge in the background, we wanted to assess to what degree the association between cone and RPE mosaics can be observed in the foveal shape that we obtain from OCT imaging. Because if the macular RPE, because of its critical role in the cone cell cycle and in supporting foveal cones, is implicit in forming and maintaining foveal shape, then we would expect to see a strong association between the distribution of cone and RPE cells and foveal shape in the adult population. So in the data I'm going to show you, we included healthy people aged from 15 to 65 years old, and we did high-resolution reflectance confocal, dark field, and split detector AOSLO imaging, and OCT imaging. And when for the OCTs, we acquired fovea-centered OCTs, and these are corrected for lateral magnification factor using a Goldstrand model that we use. And then we use a semi-automatic method for detecting the ILM and the RPE in this case that we use for more analysis. And for the foveal shape, we just did an cubic spline interpolation that we have used in the analysis. And some of you here are already familiar with multimodal AOSLO, where you can see the dark field image at the bottom there, where we can identify RPE cells, and I hope you can see them as well. And then for the confocal wavelength, we can count the cones, but we can also look and count the cones from the split detection image. So we imaged all of our participants, typically with a 1 × 1 degree field at several locations from the fovea center and out to 6 degree eccentricity. And then we identified cone and RPE cells using a semi-automatic algorithm that we have. And then for the cones, we typically counted them over 50 by 50 rows, whereas the RPE cells, we counted over 200 by 200 rows. And then we used a mixed effects model to look at the influence of cone and RPE cells on foveal shape. And this is typical of, this is an image sequence from an atypical participant, because we, in this participant, we can actually see an image RPE cells all the way out to 15 degrees. For those of you who haven't seen these images before, I'm not sure how easy it is for you to see the honeycomb pattern in the images here. And you can also see sometimes that there are some cones coming through. Typically, most people we can image using dark field and identify RPE cells within the plus minus 3 degrees, but some individuals also are out to about 6 degrees. It's very rare that we get images of the RPE this far out. And here you see from another participant from the fovea and out to 3 degrees. And I'll just show you how we do the Voronoi tessellation on top of them. And you can see here that there's an increase in RPE cell size and a decrease in density as you go from the fovea and out to about 3 degrees. So in the semi-automatic algorithm that we use for detecting these Voronoi domains, we added a step that allowed us to move the centroid of the cell around, and also we could add cells if there were, and take away cells if they weren't visible. And here I show you the geometry. So this image has gone a little bit wrong in the transition from Mac to Windows. So there's too much red here, because basically it should have been only this small red one showing, and then the rest of the red shouldn't be showing. Because this is the 50 by 50 row where we identify the cones, and this is the 200 by 200 row where we identify the RPE cells. And then you can see here how we can count how many cones there are within one RPE cell. And yeah, I think it's enough to say that. So what we did with this data was that we exchanged the data or pulled out the ICD data, the intercone distance. So for every single cone in a 50 by 50 row, we pulled out the information of the eccentricity location and the size or the intercone distance for that given cell. Because that gives us a lot more data for every single location, and it's also then independent of the size of the row. But regardless, I've given the cone and RPE cell densities for the rows in the table on the right there. And you can see like cone density that we've known since histology, that there's a change, a deep change, deep decline in cone density as you go from the fovea and out. And we see a similar decline in RPE cell density, except not so big, because the density of RPE cells isn't that large. But the other thing I think you also can appreciate that there's quite a bit of between individual variation, not only in cone density, but also in RPE cell density. And at the bottom there, I've given the cone to RPE ratio at the peak cone density position that we used. And then on the figure on the left, you can see the data for all the participants for cone density and RPE density. The figure on the right is RPE density. So you can see that that's quite a bit flatter than for cone. And then here are the ICD data. So every single cone and every single RPE cell that we counted. And then at the bottom, you can see the where we have converted the ICD data back to cone density data, assuming that they are hexagonally packed. So we use all this data for modeling. But the other thing we also do, because in our system, we cannot image the foveal cones of every single participant. And in this study, 12 of the participants, we could image the foveal cones, and the others we estimated. So we fit power functions to the cone densities and generalized exponential functions to the RPE cells. These were the best fits for the data to estimate peak cone density for those that we couldn't count the foveal cones for. And the best fit models were also asymmetric models, showing that which we know from histology as well, that the density is different on the temporal versus the nasal side. And here I've just added the peak values for these individuals. And here I show you then the number of cones per RPE cell over the central 1 degree. And this is for those 12 participants where we could image the foveal cones. So you can see here that there is quite a big variation. For some, this profile is quite peaked, and for other people, it's kind of flat. And I've given the peak cone density estimate for 3 of the participants in this figure. And the next step we did was that we looked at how the ICDs changed with the eccentricity. And we found that both cone and RPE ICDs increased, and the number of cones per RPE cells decreased with increasing retinal density. So we could use this for further modeling. And here I show you the modeling data. So here we've used the cone ICDs and we've used the RPE ICDs in a mixed effects linear model to see if we could use that to predict the foveal shape. And what the model shows is that we need both the cone ICDs and the RPE ICDs to do this with high degree of accuracy. So basically predicting the foveal shape from these 2 densities tells us that 98.5% of the variation is explained. Only 1.5% of the variation is not explained by the model. So in determining foveal shape, the RPE and the RPE you are given or develop before you're born is probably very important in terms of how your cone density and cone mosaic changes. So here I've just shown you the profiles in those T. And this is then the data for all the participants in this study. And this is, what we've done here is kind of a first step in looking into these aspects. And I've put a circle around 2 of the individuals here because their profiles look kind of very similar. But these 2 people have a very different eye length, for example. So there are many different things that we need to understand more about the consequences of the mosaic that you have. Some other results is that we saw that there was a negative correlation between linear cell density and axial length, which other people have shown before us. There was no association between foveal cell density and age, which also has been shown by Curcio et al. And we saw that there was a positive correlation between estimated peak cone and RPE cell density in the foveal center, which is kind of intuitive because it means that no RPE cell, then there are no cones. So there is a close links between these things. So we show that there's a strong relationship between cone and RPE cell spacing and the shape of the fovea. And we wonder whether the RPE play a role in how the foveal pit is formed and maintained, and putatively also perhaps imposing constraints on the number and distribution of foveal cones. So I think I'm going to stop there. It says 29 minutes here. And I guess we probably have time for 1 or 2 questions. Thank you for listening. And now I have to look here as well. You got a question?
I was interested by the modeling of the foveal shape. You published also some work about quantification of cells. And where all the shape of the fovea displayed on this slide showing the curves, all your subject were there.
Yeah.
Okay. So you have no flat fovea, no.
Where I had someone who didn't really have a foveal pit.
Yeah.
Is that what you're asking me?
Yeah.
No, not in this data set. No, that's interesting. Yeah.
Because we have a very strong, I don't know if it's related to ethnical diversity of the population, but in black subject, fovea is much larger, and blonde-haired people, it's much less. So it's you find such changes, or do you have enough subject to check on the correspondence with the cell population?
Not in this data set, but I think that is really interesting to look to actually have more subjects as we go along, and some with those much wider foveas than we have here, to see if what we found also holds true in them. And because I think that would also be important in terms of why is it that, particularly this difference between people who have a lot of pigment compared to those who have not have a lot of pigment, but still within the normal range, the difference in prevalence in AMD again. Is there something to do with how the fovea has shaped, and then how many RPE cells you have and how many cones feeds into each RPE cell in terms of the risk of developing it. Okay, one more question.
Curiosity question, but it was really interesting to relate. I never thought could relate the original thickness with the RPE and photoreceptors context like this. I was wondering also the fovea umbo,
yes,
the elevation. Why? I mean, can we? What's the origin of this?
What's the origin of the umbo?
Yeah, because the thickness, there's some relation, strong relation with the cone density and the RPE. Is that something related to that?
So I'm not quite sure what you're asking me now, but are you asking me why we have a foveal dip?
Yeah.
Okay, so that is, so through development, the cones migrate in towards the fovea center to create the fovea center. And the cells that are in the inner retina, they move to the side to make sure that what reaches the foveal pit is light, and that doesn't go through several other layers. So how the foveal pit is, or the fovea is shaped, we know from histology, but we also can see it now from OCT images. And what we also see is that, of course, a lot of things happens before you're born, but then there's a lot of things also going on from you're born till probably the end of adolescence. So cones keep packing, and the fovea develop further, all the way at least until 16 is their data.
It could be a relationship with cone density. Shape.
The cone density and the shape.
Yeah.
Well, what we showed you, the peak cone density does not correlate with foveal shape. Other people, Joe Carroll and they have looked at that. So peak cone, just the peak does not correlate with foveal shape. You need to look at how the distribution of cones is through the flat part, and then through all the way up to parafovea. Okay, then.
There's one more question if you have time.
Okay, when then Ysé can come and get ready, and then I'll answer questions. So where was the other question? Please.
In the middle of the presentation, you said that you results of the 12 participants for the outcome, and also you estimated. Did you use some special model, or you developed based on results?
So in those 12 participants, we could resolve foveal cones and counted foveal cones. So they were not estimated. For the other participants, we estimated peak cone density by using a power function. And we tested several different models, but this was the model that gave the best fit. And the best fit was also one that was asymmetric, so accounting for differences in number of cones, nasally versus temporally. Thank you. Then over to our next speaker, which is Ysé. Please, take it away.
Okay, hello everybody. So thank you for being here, and I'm happy to present my work of my first year of PhD that I'm doing in the Paris eye imaging group and more precisely in the HistoParis group. So today I will talk about this work, which is deciphering the cellular dynamics during the progression of dry AMD. So I'll first talk a bit about dry AMD. What is dry AMD? AMD stands for age macular degeneration. I think most of you have heard about it, but simply so it's a disease that affects the macula. So it's the center of the vision that you can see on the white square here. On the fundus photograph, you can see like this lesion. It's like a flower-shaped lesion standing on the fovea. The problem of this disease is that this lesion progresses along the time, and this what will affect the vision of the patient, and the patient will complain of visual loss because of the progression of this atrophy. This is atrophy of the retina and RPE layer. So on the histological part, first data was known about the histology, classical histology with a slice section. You can see that in a normal patient, like the RPE layer is not affected at all. You can see on this image here, whereas on dry AMD patient, the RPE layer is really affected. It can go from the beginning of AMD to a disorganized layer of RPE cells with cells that can, the layer can be doubled, or even to the dry atrophy lesion, you have nearly no RPE cells inside the atrophy. So you have up the little schematic. So you can go from an healthy RPE layer to early AMD, which is composed of drusen with the materials coming under the RPE layer. And there are several hypotheses about the coming up of the atrophy. One of them is epithelial mesenchymal transition of the RPE cells, which lose their epithelial characteristics, going and then coming to atrophy. This is not really the hypothesis we support, but we will try to demonstrate that this is not what happens in reality, but it's a matter of debate. So what we can see from the ophthalmologist point of view, what we can see on the exams we're doing on people. So we are doing infrared imaging, and you can see on the infrared imaging that the lesion is increasing across the time. You can see also, and then from the late 90s years, you can we have the OCT, which is currently used for the follow-up of the patients, and you can see also the large atrophy that yes, I have to here. This is the atrophy, you can see that the hyper-reflective line here is the RPE line. You can see it completely disappeared, and this line increased across the time. So what do we get interested in AMD? In fact, in AMD to date, there is absolutely no treatment to prevent the inhibition or the progression of this atrophic lesion, resulting to the poor vision in the patients. But there are no treatments because the mechanism that underlie the progression and the initiation of those lesions are really not known at all, and the main hypotheses are not shared between all the experts. So the purpose of our work was to characterize the dry AMD at the highest precision scale, and this combining two different techniques, which is the in vivo imaging with high modality multimodal resolution imaging and the histology of the RPE, to combine in patients the dynamic of the RPE cells and the atrophy, the growing of the atrophy, and on the histology part, the cellular and the molecular cell resolution scale. So why can we do that? We can do that because I think that most of you have heard about the clinical imaging lab, and most of you have visited it already. So we can do this because thanks to the large diversity of multimodal imaging in the lab. And so what can we do with this? We can see the progression of early AMD with something with adaptive optics and a technique called gaze-dependent imaging. We can see small drusen even in patients where you cannot see anything on the color fundus on the photograph or on the OCT. So with this kind of technology, you can see really small drusens, and you can even follow them in the time. This is a time lapse showing the beginning of the lesion where it is traced here to the expansion and the growing of the drusen. And it's the same for this one, but for all the drusens that appear on this image during a 3 years time lapse. But we can do also the same for AMD with dry AMD patients having atrophic lesion. So you can have a cell resolution imaging with adaptive optics ophthalmoscopy, and you also have the time lapse showing the evolution and the growth of the lesion on the infrared imaging here. And you can also combine both to have the cell resolution imaging and the time lapse imaging to have the dynamic in the patient on the growing of the lesion. So you can see that nearly at the cell resolution imaging, the growing of the lesion with the atrophy on this side and the RPE retracting on the other side. What is interesting on these images is not only the border that gets rid of the atrophy, it's also all that small black points that move a lot. And so we think that it's pigment. And what is interesting is that this pigment is highly dynamic across the time. So we get also interested in pigments. So why pigment? Because pigment is really a hallmark of AMD. In every AMD patient, you have a redistribution of pigment clinically. So it can be interesting to have some techniques to image it. It has also been shown that it's accumulated in the inner retina, but in inner cells. So that can, in fact, we have like a cell marker, which is like a cell tattoo that can be imaged with in vivo imaging techniques and without labeling in patients. So that's really interesting to visualize the dynamics of probably macrophages, which have like eaten the pigment, the melanin debris, and which move across the time. We have different type of pigments. We have the pigment located in the inner retina. And what's interesting in this type of pigments, called hyperreflective foci, is that this pigment doesn't move across the time. You can see that the border of the atrophy is moving and is retracting, whereas this pigment doesn't move at all. And you have also that kind of pigment which is located on the border, on the RPE layer, and under the retina, which is moving with the retraction of the border. And this is really interesting. You can image the pigment by different way today with the near-infrared autofluorescence, because it is thought that melanin is autofluorescent in near-infrared. You can see here this pigment autofluorescent here, and you have the correlation with adaptive optics under the form of these black points being pigment. So we are trying to develop new methods for imaging pigments in patients across the time. One of them is scleral slab. So what is scleral slab? It is done from an OCT en face imaging. Currently, it's done with the plexelite. And from this, you're doing a section really profoundly, deeply under the choroid here, RPE and retina in sclera. So you have a scleral section reporting all the hypertransmission or hypotransmission from the OCT signal from the retina and RPE. And what we saw is that from the B-scan OCT, we saw that in fact the hypotransmission corresponds to pigments and hypertransmission correspond to defects either in the RPE layer or some small cavernas in the retina. So here you can see, for example, on this scleral slab, this large hypertransmission corresponds to the atrophy, whereas here all the point of hypotransmission corresponds to pigments across on the RPE layer. So what's interesting is that we can also do a time lapse of those images, and it can give you across the time a dynamic map in 2D of really a large field of view of all the pigment redistribution across the time. Another method more at the cellular scale is the TFI, which has been presented, I think it was on Wednesday by Elena Goffas, which is an imaging method through the sclera. And you can get the signal through the pupilla. It was designed to observe the RPE mosaic in vivo, but we found that we can see really well the pigment with it. So here you have a multimodal imaging of the pigment in one patient. Here you have a pigment, some pigment. You can see here in a near infrared autofluorescence that it's autofluorescence. On the OCT, you have a small hyper-reflective here spot. Thank you. You can see also here in adaptive optics, and more precisely in you can see really small, nearly at the cell scale level, the pigment on the TFI. So that could lead us to map really pigment in a multimodal imaging with all the techniques we have. Whereas we cannot really see it on the fundus photograph because it's really, really small, and we can see it with many different imaging techniques. We can go further with the AO OCT. Here you can see even the defect on the RPE layer and the pigment probably coming from this RPE layer. The scleral slab, as I told you before, and cellular resolution scale with TFI. So, and with this, what can we do next? We would like to do histology to document all those changes at the cellular and molecular level. So how do we do that? We use human donor eye. They are preserved in formaldehyde in -80 degrees for the treatment of the tissues. Then we defrost them. We treat them like we do for clinical patients, and then we stain them for histology. So this looks like this. So this is a patient who had an AMD, and you have the piece of interest with the sclera here, and RPE choroid is this layer, and retina has been put apart, and you can see the atrophic zone here. We do then clinical OCT. We use the same clinical device that we use for patients. So you have here the smaller mounting to image the sample. And then we can do also blue autofluorescence and near-infrared autofluorescence, and also OCT on our pieces of interest to correlate them with what we observed in clinics. So first of all, what looks like an aging RPE. We can stain really nicely the border and the cell structure of the RPE cells with phalloidine and staining the actin to have the morphology of the RPE layer. And then we can correlate this with the Norberg optics that give us the pigment loading of every cell. And that's really interesting because, as I told you just before, pigment redistribution in AMD patients is really important. We can say that in 90-year-old patients, the RPE layer is not that bad, in fact. The cells, most of the cells have a good shape, but some are a bit swallowed, and the pigment redistribution is, however, very altered with nearly white depigmented cells, some being much more pigmented. So really pigment is really differently reparted in the cells during aging. And when we begin with histology on small atrophy, it can give this. So here you can see in blue autofluorescence, you have here the dark spot representing the atrophy here, and in near autofluorescence autofluorescence, you also have this hyperautofluorescent spot with slightly hyperautofluorescence on the border of the atrophy, probably coming from the melanin accumulation on the border of atrophy. You can see the corresponding OCT, and the atrophy is here. You have the stop of the RP layer here and here, corresponding to what we can see on these pieces. So on these pieces, on the photograph, you have the retina on this one. You can see here the atrophy and small drusen's around. I'm not sure it's clear on the projector. And on the RPE, when you can remove the retina, you can see clearly the atrophy, the atrophic lesion on the RPE. And when you stain it with you can do a multi-labeling on this piece of histology. You go through this, so you can do really large field of view, and this is really interesting. It takes a really long time to acquire in confocal microscopy, but I hope it's worth it. And when you can also zoom in on the border to see the cellular structure of the RPE layer here. In red, it's the RPE 65, which is a marker of the RPE activity and marker of the RPE cells. What's interesting is that in green, it's the inflammatory cells, it's IBA1, it's a marker for inflammatory cells. It's that inflammatory cells are not many of them on the atrophy, and this is interesting. So what is, we don't really know if it's really inflammation that triggers the initiation and the progression of atrophy. Thanks to the Norberg optics, you can do also the phase contrast, and you have in the phase contrast all the pigment distribution and redistribution in all the cells surrounding the atrophy. And you can see that it's really not regular, and so there are really pigmentary changes during the evolution of AMD. Here you can see it is the fellow eye. On this side, it is interesting because the atrophy is not complete, but so we are located in the fovea. Atrophy is not complete because we still see RPE cells, but they are really deformed, but they still have the shape because they have their links with the other cells. And what's interesting is that the shape is really messed up, in fact, even if they are still there. So atrophy is not complete, but there is something preliminary to atrophy leading to rarefaction of RPE cells, but the remaining RPE cells keep a good shape, and probably they keep their epithelial characteristics. When you have the final evolution is the large geographic atrophy. It can give that kind, so it's the pieces I showed you before. On the blue autofluorescence imaging, it gives here you have the atrophy, which appears hypoautofluorescence. This is all those is tissue. It's the tissue which suffer a bit from the preservation, but anyway. And when you do the OCT, you have here the stop of the RPE layer, and you can even see small toxins standing inside the geographic atrophy zone. And when you do the histology on it, you can see this. So you have really a beautiful RPE cell layer with RPE cells that seems really epithelial, keep their polarity, keep their intercell bridges. When you go not far away from the border, you can see that the cells seems normal, like in the 36-year-old patients. On the border is the same. The cells doesn't seem really harmed. They seem a bit compressed, as if they were compressed, but that's all. They're not really dysmorphic, and they look nice, in fact. And there is not much inflammation. Inflammation is seen as red. It's the red cells. There are a few of them here and here, and that's all. So you have a strange region of interest with RPE cells looking a bit bad, a bit worse than the other side. And what's interesting is that it's the part of the cells which are depigmented. So there are also alteration of pigments in those parts. Whereas on the border, the cells seems nice, and it's even more blacker than the other cells. So probably because of the compression of the cells. Here is another example. It's the fellow eye. So you have the same characteristics with large cell here, but for the other cells, it's normal. And same for the pigment redistribution. And if I can go further, we can go further and do zoom in this RPE layer, and you can see here this bunch of cells that's still tied together and not really not messy, and they're still here, even if the shape is not the traditional cellular hexagonal shape. And so I will end on this final, which is another final slide, which is another example of geographic atrophy with also the multimodal imaging of the segment that we can correlate well with the imaging we do in patients. And here you have the atrophic zone. You can see here with absolutely no RPE cells which are stained which are labeled with the RPE 65 in red. Here the OCT, blue autofluorescence, near-infrared autofluorescence with again this hyper signal on the border of atrophy and the corresponding OCT. So this is I think really the strength of our team. It's the possibility to do really something translational for the study of dry AMD, and 3D histology is really interesting for the study of the cell mechanics during initiation and progression of atrophy, and we can in that way understand the cell interactions between different cell types, between the RPE cells with the photoreceptor, with the macrophages, etc. We can try to stain different biomarkers characterizing the RPE cells themselves. And we would like to go further in the pigment imaging and the RPE cells imaging in vivo in AMD patients by collaborating with the Ethan Rossi team in Pittsburgh. And so on this, I would like to thank you for your presence. And I'd like to thank all the members of the team, mostly the Histo-Paris team and all the Paris eye imaging group.
Thank you so much for a great talk. Are there any questions? I think there's 2 questions maybe. Okay, go on.
Yeah, you mentioned the dynamics of the pigments.
Yeah.
What's the time scale actually? Yeah.
For the time scale, it's the same as AMD. It's like three years, and usually we took images between 3 to 6 months.
Okay.
So maybe we should.
Would you be were able to track pigments movement?
No, I did not. I
Maybe
didn't.
they're faster.
Yeah, we are tracking it from. We did not trace it. We can, if you look, if we can track the pigment variation, but I didn't do it like manually.
There are 2 questions. Do you want to read them?
Does this work only on swept source OCT? I haven't tried on the other one, so I cannot tell. I tried, it works well. The OCT I used there is a top con, and it works also on the Heidelberg, but I didn't try for other OCTs. Cholesterol accumulations at the level of RPE also creates artifact on sweat source OCT. Do you know how does it behave when you perform scleral slab? Probably. It also creates, yeah, because there are also the calcium, some dozens are calcic, and we do not take into account the calcic part or the cholesterol part of the dozen in the scleral slab. It's true that it can do an artifact on the scleral slab, yeah. So we have to, for the validation of the method, we have to correct the B scan to see if it's really due to pigment or other artifacts.
Thank you so much. Thank you, Ysé.
Thank you.
And then the next speaker is Gemma, please. Do you want to come up? Try to find you.
Hello, everybody. Yes, so as Rigor said, my name is Gemma, and I'm going to talk today about the Belfast Eye and Multiple Sclerosis study. So first of all, I'm going to absolutely apologise on the whole of Belfast on my accent, because yeah, I'm going to try and speak as slow and as clearly as I can, but we are probably the worst place in the world for our accents. But here goes. So if you didn't guess from the name, I'm going to be talking about multiple sclerosis. So it's a progressive disease of the central nervous system, and it's characterized by 4 main pathologies. So inflammation, glycosis, demyelination, and neurodegeneration. And as you can see in this picture here, it's actually this protective myelin sheath that wraps around the neuronal axon that becomes damaged due to being targeted by autoantibodies. And much just like your phone charger, the insulative wrapping that goes around it, if you leave axons bare or the wires in your phone charger bare, they're eventually going to die. So unfortunately, this does lead to irreversible neurodegeneration. And unfortunately, at this stage in time, the diagnosis and monitoring of disease is currently by invasive techniques. And this is only available after that irreversible damage is done. And I'm going to talk about some of the reasons as to why that happens. But first of all, I'm going to explain a little bit about the types of MS. So the most common type of MS is called relapse remitting MS, where patients go through stages of these relapses here, as you can see in the blue line, where disability tends to increase. And then they recover as to where remyelination will occur, so that protective wrapping wraps around the axons again and protects them from the damage. But unfortunately, this happens over and over again, and patients can go up to 10 years or at least a couple of years, but some unfortunately do go 10 years before being diagnosed. And then eventually around 15% of patients will progress to secondary progressive MS, where they don't really go through any relapses anymore, but their progression continues linearity. And so the problem with this is that because patients recover, by the time they get MRI scans or invasive techniques, they've recovered, those demyelinated plaques are gone and we can't pick them up. All the while the disease is progressing and we can't do anything about it. So thus comes the concept of the eye as the window to the brain, which I'm sure all of you are very, very well versed in by this time. But just in brief, so the brain and the retina share many, many similarities. The first is that they're from exactly the same embryological origin layers, so the developing diencephalon. They're obviously structurally very similar. They both contain many layers of neurons, just like the retinal ganglion cell here, as you can see. And the axons of these go then into the optic nerve and into the brain. And the optic nerve is myelinated, so much like the brain. However, as you all know, the retina is not myelinated. And so you're probably thinking, why would MS affect the eye if there's no myelin in the eye? But that's quite controversial, so I'll leave that to everyone else. And they also share other similarities, so microvascular structure is very similar, and they're also both immune privileged sites. So what do we already know about MS in the eye? Well, we know that 90 to 95% of patients will have some kind of ocular pathology throughout their disease course. And the most common one of these is optic neuritis, which has been spoke about as well before today. But in brief as well, this leads to optic nerve demyelination and degeneration, which then in turn leads to inner retinal degeneration, so retinal ganglion cell layer and the inflexiform layer. And we also see decreased vessel density, which has been seen to correlate with cerebral hyperperfusion. And also then functional deficits, so in our best correlated visual acuity and decreased contrast sensitivity. But most importantly, what seems to be quite promising is that this retinal degeneration does seem to correlate with brain degeneration. And so us in little tiny Belfast decided that we would try during COVID. Well, we tried before COVID and then COVID came, and unfortunately we only got around 16 patients and 24 controls. So it starting off as a pilot study, an explorative and feasibility study, and we put participants through rigorous tests. So they were with us for around 3 hours. They went through fundus, optical range tomography, adaptive optics, ultra wide field imaging, OCTA, habitual visual acuity, contrast sensitivity, and Goldman wide field perimetry. So they also had other tests done as well, but I'm not going to speak about it. We did MPOD, multicolor, quantitative autofluorescence, and IOP and different things as well. But just for the talk today, I'm going to talk about OCT, adaptive optics, and then these two functional exams just to correlate. So first of all of our demographic information from patients was obtained through our consultant neurologist, so Dr. Gavin McDonnell and Dr. Stella Hughes. For our healthy participants, it was just through a health questionnaire. We didn't have any records of their GP or anything. So there was no significant difference in age or gender. And there was no significant difference between disability scores or disease duration. And we also looked at hypertensive medication because we also know that this has an effect on the eye. So first of all, OCT. So we use the Heidelberg Spectralis, and we use this Insight module. So you can see here on the left, and I just include this in case anyone wasn't familiar, but you probably all are. So the default retinal module starts the scan basically on the temporal side of the retina, which is exactly what we're interested in. And so whenever it finishes back around to this temporal area, it's not quite as seamless of a measurement. So then they created this new Insight module specifically for neurodegenerative diseases, where they start on the nasal side and then get quite a seamless measurement then of the temporal side. And it allows us then to measure this peripapular macular bundle, or this PMB measurement, which is basically a very, very important bundle of fibers that goes right from the fovea into the optic nerve and into the brain. And it seems to be quite affected in neurodegenerative diseases. And then we also get this nasal temporal ratio as well. And then our macular scans, where the posterior pole scans as well using the Insight module. And what it does is it allows us to then angle the scan right over this peripapular macular bundle. And at the start, we were just looking at these inner retinal layers because that was what was previously seen. And what we wanted to do, because we had such a small cohort, was just to confirm if our study did in fact agree with previous studies, to try and gain some trust in our database. So thankfully, globally, so I'll just go back 2 seconds, I forgot to mention. So global measurements will be all of these sectors averaged. And then furthermore, because of the neurodegenerative aspect of this, I looked more at the peripapular macular bundle and then the nasal temporal ratio. So we only find a significant thinning or degeneration in the retinal nerve fiber layer in patients with a history of optic neuritis. Oh, sorry. So MSON means patient's eyes with a history of optic neuritis, and then MSNON is eyes without a history of optic neuritis. And we didn't have any optic neuritis in our controls. And this is quite common in the literature, see. But then actually when you look into each individual sector, within that peripapular macular bundle, we actually see that this thinning or degeneration is independent of optic neuritis. And that was then followed up in the nasal temporal ratio. So just for easy visualization, as you can see in patients with a history of optic neuritis, there's quite this global kind of damage around the whole optic disc. Not surprising, but in patients without optic neuritis, there's still a thinning in this peripapular macular bundle. And so then we moved on to the inner retina, and we found a significant decrease again in only those eyes with a history of optic neuritis. This was in the ganglion cell inner plexiform layers, and then a trend towards thinning within the inner retina, most likely due to our numbers. What was quite interesting is that when we looked at the retina thickness as a whole, is that we didn't see that thinning anymore. And this kind of sparked interest into, is the outer retina affected? And as you all probably know, is that thickening of the retina is most likely indicative of inflammation. So we wondered if this could be playing a role. And so when we looked at the outer nuclear layer, so the cell bodies of those photoreceptors, we didn't find any difference between the thickness of these layers. However, when we looked at the photoreceptor layer, so those layers only including the inner and outer segments, we actually find an increase, but again, only in those patients with a history of optic neuritis. And this held true then when we compared the outer retina as a whole. So this went from the bottom of the outer nuclear layer down then to the RPE. So then just to make sure that the RPE wasn't affecting any of these measurements, we see no difference in the RPE when we measured them. So thus starts our story with adaptive optics. We then wanted to know if the cones then were affected in MS. And so you all know this schema. I mean, you have all presented very, very, very cool pictures. And then I've got my little like primary school drawing. But basically, yeah, we're able to view the outer segments of the retina using adaptive optics using a resolution of 1 to 2 microns compared to the OCT, which is a resolution of around 7 microns. And so what we did was we imaged at 0 degrees over the fovea right out to 6 degrees temporally. So with the images, it allowed us to view up to 8 degrees temporally. And this is what the adaptive optics screen looks like in the RTX1. And what we did is we semi-automatically segmented cones. So this is just the automatic segmentation, and then I would have manually went in and corrected some of these, or all of them. And then I first of all wanted to look back to see what I would expect from a graph of cone density. And as you can see, as you all know, there's a very, very, very high increase, or very, very high density of cones right at the fovea, which has a non-linear, extremely steep slope out to the periphery. And so we did find significant decrease actually in cone density in patients with MS. And this accounted for 10 to 15% of cone loss. And this was then followed up then by an increase in cone spacing as well. And so because of the difference between optic neuritis history and OCT scans, we looked at whether this was causing an effect in the patients. But actually, when we looked at it, all our p-values were 0.99999 between patients' eyes with and without a history of optic neuritis. So this cone loss is actually independent of optic neuritis. And then furthermore, just to see what functionally these patients' eyes were doing, we see a trend for a decreased visual acuity using the Snellen charts. But in contrast sensitivity, this then proved significant only in those patients with a history of optic neuritis. So even though they seem to be either losing cones or just have less cones, we're not too sure. We can't really tell from our data. Their functionality still seems to be okay. And so in summary, our very small cohort did seem to agree with previous studies of retinal degeneration. And we do see a significant thickening in the outer retina, or specifically the photoreceptor layer. We see a decreased cone density in patients with MS that does seem to be independent of optic neuritis. So then our next steps are to follow up on the vascular analysis, because there's been really, really cool stuff being presented here today. So we're currently doing a fundus automorph. It's quite similar to the SIVA analysis. We're also looking at our ultra-wide field vascular analysis using the Vampire software from Edinburgh. We've got our OCTA data analysed, and then we're going to look at the choroidal thickness and choroidovascular interest index. And then we obviously want to roll this out into a much, much larger longitudinal study to follow up with these patients. So just to acknowledge everyone that has helped me got here, shout out to my supervisors, and obviously the Imagine Eyes guys as well, and my funders are Optos. And that's it. Thank you very much.
Thank you, Gemma. Wonderful talk. You don't have to make excuses for anything. You did really well.
Thank you.
Are there any questions in the audience? There's one, please.
Did you already try to compare with the visual field or some functional test and the reception of like acute phase? Sometimes, and especially suddenly, decrease the visual acuity and the visual field. So in such case, did you find any correlation? Or?
Do you mean in optic neuritis or?
Yes.
Yes. No. So all of.
You can repeat the question.
Sorry.
And
so the question was whether during optic neuritis they have a massive deficit in their visual acuity at the time, and did we try and correlate that with cone loss or anything? But we actually, because of the impact of optic neuritis has on the retina and OCT findings and everything, we excluded all patients that had optic neuritis within 6 months. So we didn't get to look at that, but that would be extremely interesting. That would be very cool. No problem. Thank you.
Yep.
Are there any
questions?
Oh,
Here?
there actually might be.
Yeah, there's one. Do you want to read it?
Yes, so the question is, any functional abnormality related to the enlarge of RPE cells? So we didn't actually get to look at the RPE cells. My supervisor has, did I get that right? Is that what that means?
Yes, that is, but I'm wondering whether that was,
maybe for you,
it was a late question for Ysé or me, actually, although it's the last question. So we'll leave it.
Okay, we'll leave it.
Thank you. Thank you so much, Gemma.
No problem. Thank you.
So let's see, the next speaker should be on Zoom. I'm just gonna try to close here. So the next speaker is Katarina, I think.
So hello, thank you. I'm not sure if you can hear me.
We can hear you. Do you want to share, please?
I'm sharing and
Okay.
so is it fine for you? You see my screen?
Hang on a second, Katarina. We just need to move your screen to our.
What is this? Yes, I need to, I have to, yes. I think it's just, yes, I can troubleshoot. This one. From the call, and then we can remove. No, this the other side. Yeah, and you make it larger. Yes.
Is the sharing not available? Yes.
Yeah, it's
And then just full screen.
and then you
Just pull it to the side. You can just take the line.
Yes.
Is that okay? Then Katarina, please start.
Okay, thank you very much. So I'm very happy to join you through the internet today, and I will present you a project that we did with patients with choroideremia and adaptive optics imaging. We used the RTX device, Imagine Eyes, and the purpose of this project was to analyze the morphological retinal patterns and parameters in eyes with choroideremia. It was a project combined with observational trial of patients after gene therapy on one eye, and also additionally we analyzed vessels of these patients if there are any changes observable after the treatment in the sense of immunologic reaction or just any changes you could observe on the vessels. So for those who are not familiar with rare eye diseases, choroideremia is an X-linked disease that is leads to genetic degeneration of the photoreceptors and the choriocapillaris layer, and then even more pronounced than retinitis pigmentosa and other photoreceptor diseases, you see basically the scleral tissue in areas where the degeneration is complete. In the middle of the retina, in the foveal and perifoveal region, usually the remnants of tissue are until a late stage of the diseases, and the progression of the disease is similar like in retinitis pigmentosa and other peripheral disease, progressing towards the middle. In long-term progression, choroideremia leads to blindness as well. We included in this short project 5 patients with choroideremia, and those ones have been treated with gene therapy prior to this project on one eye. Probably you know there have been trials with gene therapy for choroideremia, and some patients even received the treatment on both eyes. But here we only included volunteers with one eye treated, and because this project has been supported by the Orphan Disease Center in University of Pennsylvania for 1 year in 2020. There were no patients before and after treatment, so we only compared the treated and the non-treated eye, so we don't have images of the same eye before and after therapy. Here the comparison was between the 2 eyes, and all the patients have been in the time course 12 or 24 months after they have received the genetic therapy in the one eye. We used combination image of the RTX1 camera of 17 single images. This is then covering approximately 13 times 13 degrees of visual field. This is the macular region. And then additionally, we made an montage image of 5 overlapping images on the upper arteries, on the vessels, and this was used for the vessel evaluation. So you probably know the combining images, like it looks in a normal eye. The alignment works very well, and on the left side is typical patients with choroideremia. And of course, the alignment works as well, but the image quality is very different. So to start with problems, sure, there have been differences between normals and these eyes. And the first thing is bad fixation, because patients have quite good visual acuity until the end, but these included in the genetic therapy were rather progressed, and this influenced the image quality. We have seen that there are only small remnant and intact areas of the retina, and due to this combined generation of the photoreceptors and their underlying vessel layer, there is a very different reflectivity. This loss of the retinal pigment epithelium is very specific here, and I will explain you later the patterns we have seen, and one of them is a little bit depicted here on the right image. This like flat sensitivity, no structures, and here the RP is missing, and what you see if you compare it to the classical retinal imaging, then there is a complete loss of the tissue up to the sclera. So here a little bit enlarged, and this is an image captured between the retinal vessels, as you can see. So if you combine these images to the bigger picture, of course, you can align or compare it with the other types of retinal imaging. And here you see an autofluorescence image, where, of course, this contrast between preserved retinal tissues and the other areas is much bigger due to the reflectance of the pigment epithelium and of the fluorescence imaging. But still, even in optics, the edges of this atrophy are very well visible and identical with the other kinds of imaging. So sorry, this doesn't click further. Oh, that was too much. Let's go back. Here you see a comparison not only to autofluorescence imaging here on the very left side, also to infrared imaging, and of course that corresponds to this late stage preserved area in the middle, and also the very well visible choroidal vessels that are still available are observable on all other modalities. What about the actual photoreceptors? You can search for them only in the remnant areas in the middle, and the pattern is very different from what we know. Of course, in a normal, there are only few of them being reflective, and sometimes you don't know, are these really cones or is this some artifactual reflectance? But if the patient has a good fixation, you can see some preserved areas of the typical photoreceptor mosaic. A next pattern that we have observed have been these RPA clumps. They exist in the retinas in the process of degeneration. And this is exactly what I'm talking about, these small areas. And if you enlarge them, then you can also search for them in the other modalities. And here, autofluorescence imaging, of course, is not really useful because this doesn't give a contrast. Here is much better in the infrared imaging, or even the best in adaptive optics, because you can define these edges inside of the outer retina. So far, I have described patterns that have been visible in infrared imaging and autofluorescence imaging, but there are also aspects that can be seen and have been seen by us only in adaptive optics imaging, and was in the beginning difficult to understand what they are actually meaning. And what I'm talking about are these bubble-like structures. They are unique in choroideremia. We have been examining with our TX1 for, I think, 5 years now in our special clinics for inherited retinal diseases, and we have about 200 images from RP patient, Usher syndrome, cone dystrophy, Stargardt's, and so on. But these bubble-like structures are really unique for choroideremia, or we have not seen them in other cases. So what is this about? It looks like an empty space that are present inside of the macular region. They are not inside of the preserved retinal tissue, and they are scattered all around the middle, if I may say like that. Comparison to other modalities shows then, if you also include the OCT, that here on these places actually the residues of the choroidal vessels are, and very probably because of the change reflectancy, this is exactly how the choroid spaces look like if there is no inhibiting photoreceptor layers above or the RPE. So comparing yeah, and the slide comes. Comparing this to OCT, a little bit more overview. If you put the line, which would be here, I don't know if you see my pointer, but it would be somewhere below the black missing rectangle. Here, putting it through the image, you see that the line goes through an area where there is still some remaining choroidal tissue or choriocapillaris tissue, whereas on the side of the bubble-like structures, basically only sclera is staying with the inner retina, and these are the structures that do have this very change reflectancy, as I explained earlier. So here, just to sum up what I wanted to say, we observed new structures, these bubble-like structures unique to choroideremia, describing most probably areas of degenerating choroidal tissue that we are able to see without inhibiting retinal tissue in these patients. Okay, if we observe this on a classical photography of the retina, then you also see that there are these little bit or orange reddish parts that characterize the blood choroid tissue, but you wouldn't be able to define it like this specific part without OCT, of course. And then we have asked ourselves, are there any differences on the treated and non-treated eyes? And as I said, we don't have the baseline versus after the treatment comparison, but still we have this comparison between the eyes. And of course, this comparison is not quite fine because you usually treat the worst eye, so the eyes are not 100% identical, but there is some symmetry between the eyes, and we compared in these 5 patients. Usually it was difficult to see some aurelial structures to count the cones. This is almost impossible. But there was a patient who really had, surprisingly, because you usually treat the worst eye, surprisingly had preserved structures of the photoreceptor in the treated area, and the other eye did not have it. And the question is whether this evolved after the treatment. Of course, we do not know, but it is just an observation. In other patients, you can't say with a certainty that there are some cone structures, even if they are scattered inside of the atrophy. And this is unfortunately the problem of patients with retinal dystrophies, because the image quality here is very, very different than in a healthy eye. And last but not least, let's have a look at the vessels. And on the vessels, there is a quite a well-established description of the wall-to-lumen ratio that can be established with the optics imaging. And we use these overlapping 5 images of the upper retinal arteries, and the thickness have been measured with the device. If there is an inflammation in the eye, like for example, after this adeno-associated viral vector gene therapy, we might have seen some thickening or change of the wall, sheeting of the vessel wall, and these were the aspects we have been searching for. This is how the analysis looked like, so measured at several points, and you probably know the built-in ability of the RTX device to establish the vessel wall thickness, and we measured it several times and then averaged these values for one patient, receiving then an wall-to-lumen ratio for the treated eye and for the not treated eye for each of them. Usually, and we know this from hypertension studies, the wall lumen ratio should be under 0.3, but this doesn't count anyway for patients with inherited retinal diseases because they do have changes of the retinal vessels, and usually this number is not valid for this population, but we search for symmetry between the eyes, as I said, measured at 4 different positions and averaged. The typical wall to lumen ratio in this patient is around 0.6, a little more, a little bit less. And here, okay, and here is an example of a healthy eye, which I described earlier. So example of the results in patients in treated and non-treated eyes as an overview, but more importantly here in the table, we have seen in these 4 measured patients that the averaged wall to lumen ratios are almost identical in treated and non-treated eyes, and also with more qualitative observation of the vessels as shown here, you cannot really distinguish the vessels of a genetically treated eye versus of vessels with an eye that did not receive the treatment. Peritis also showed no significant difference. And in the conclusions, we do not see significant differences in the eyes that have received treatment and that have not received treatment regarding inflammation reactions. Regarding remnant cones, the message is quite difficult because the image quality is quite low, but there is one patient, as I mentioned, that had a bit better reflectivity. What does it mean? It can be discussed. And regardless of the treatment effects, we could observe these hyperreflective bubble-like spots that are unique for choroideremia as we know it. Analysis of the volume and ratio didn't prove significant. And with this, I would thank you for your attention. And of course, I'm happy to take questions. But I would ask you to come to the microphone because otherwise, while listening to the to the course, I didn't understand very well from the audience.
So thank you so much, Katarina, for a very nice talk. Any questions from the physical audience? No questions? Please, do you want to come? Yeah.
Hi, Katarina. It's Michel Paques. Very impressive work as usual. Of course, there are many questions I would like to discuss with you about the interpretation of the image. It's fascinating to see all these different features. But you were specifically mentioning vessels. So yeah, I agree that vessels in degenerative disease are not normal. Thickened, but have you noticed any change related to gene therapy itself, which may also cause additional damage, and especially to the veins? Have you noted any inflammation around veins?
No, we haven't. And we examine with adaptive optics imaging also Luxturna patients before and after. We have also adaptive optics included in the other gene treatment trials like PD6A, but we have never seen vessel changes. And of course, all patients take prednisolone perioperatively. This is quite common in AAV vector-based trials, but no, we have not seen vessel changes indeed.
Okay, so that's very reassuring for the security of the treatment indeed. So that's, and also talking about pigment clumps, have you follow-up data on these pigment clumps? Maybe you showed it, but I missed it. I mean, does it change in shape? Does it evolve? And does that may mean something about the underlying way that RP degenerates or indicator of cell inflammation? What are your thoughts about that?
Well, we were impressed by your observation like this in AMD, and we always tried to make some visits like this over time. But to be honest, we never have been able to show it nicely as you did for AMD. So probably either the degeneracy is so slow that then we gave up, and because we didn't see any movement, or we observed wrong flags. I don't know, but we never achieved these nice movies as you have been able to show for AMD, unfortunately.
Okay, thank you.
Thank you.
I'm just going to check if there are any questions. You can see that yourself, maybe. Yeah, I don't think there are any questions. So thank you so much, Katarina.
Thank you very much for having me, and enjoy the course in vivo over there. Thank you very much.
So the next speaker is Ethan, and he's also going to be on Zoom, and he's ready. Please take it away, Ethan.
Thank you. I wish I was there in person with you all, but excited to join over Zoom here and share some fairly recent work in my lab. So I'm going to be talking about imaging inflammation in patients. But before I do that, I'm going to start with a little bit of an introduction to sort of get us all on the same page, because I know that everyone's coming from different backgrounds here, and we have a nice mix of participants. So I'm going to start with just sort of a little bit of review of what microglia are and what their role is. So microglia are these resident immune cells of the retina and the central nervous system. They're essential for both vision in mature retina and for development. As part of development, they actively prune and sculpt the retinal cells in their circuitry. And then in mature retinas, they're essential for maintaining the synapses in the plexiform layers. So if you deplete all the microglia and you remove them from the retina, as has been done in mouse studies, you don't have retinal degeneration occur, but visual function stops. And so they're essential for healthy, normal visual function. In healthy retina, microglia exist in a fairly regularly ordered and ramified state. And what is meant by ramified is that they have these processes and this dendritic morphology where they appear to have these processes extended from the soma that are very dynamic and constantly survey the local microenvironment. In primates, they have their highest densities in those plexiform layers, and a smaller proportion reside within the ganglion cell layer, and then an even smaller proportion reside on the retinal surface at the retinal vitreous interface, and those are called hyelocytes. And this is just some images from primate, from macaque, showing the density of microglia in the different areas. So you can see that they have the highest density in the inner plexiform layer near the fovea, and completely tile the retina. So these ramified microglia form a network of these potential immunoeffector cells that can become activated during inflammation. And depending on the stimulus, they can begin to exhibit a wide range of actions and roles and respond to pathogens and tissue trauma. Schematically, the cartoon in the upper right sort of shows you this transformation from ramified to this amoeboid state, where they can play both neuroprotective and neurotoxic roles. And below here is just some images from histology of what microglia from a macaque brain look like, transitioning from that resting state to the activated state. So this broad role of microglia allows them to drive acute inflammatory responses and regulate adaptive responses, and they can also help recruit infiltrating monocytes and neutrophils, macrophages into the eye when there's a break in the blood-retina barrier. So obviously microglia are important for normal visual function, but they also play a really important role in numerous retinal diseases. And they've become more studied, it seems, over the more recently, and have gathered increased attention in a number of conditions. So I'm gonna now sort of take you through a brief history of non-confocal human imaging, focusing on AOSLO and focusing on human here. So I'm not going to go into the animal studies. I will mention one of them later that is highly relevant for this work, but I just, again, want to sort of get us all on the same page here. I know for those of us that participated in Monday's session, we had some really great talks from both Elena and from Marcia from PSI. So some of this is review for those of you that were there, but again, I just want to sort of go through this and take you through the history. So the first paper on offset aperture imaging by Toko Chui when she was in Steve Burns' lab demonstrated the potential for this non-confocal imaging to allow us to see structures that we could not in the confocal image. And here the emphasis is on the structure of the vascular walls that can be seen in this offset mode. It's important to note that this was not this was the first time done with adaptive optics, but this was not the first time this type of imaging had been done. This is a figure from a paper from 1994 from Anna Elsner that shows what this indirect mode of imaging looks like for SLO. So people did this previously and demonstrated that you could see different structures. And so all the work since then is sort of built on some of these findings, borrowing now techniques from microscopy and adapting them for in vivo imaging. The work from Drew Scholes with Algebra was highly impactful, demonstrating the split detector technique here now where an ALF's instruments and many of those that have been cloned elsewhere, there is an annular mirror that picks off the confocal light for a first detector, and then the outside remaining light is split to two detectors, and then those images can be combined to further enhance the phase contrast and cancel the absorptive contrast. We sort of combined these approaches when I was at Rochester in what we called multi-offset imaging, and that had allowed us to see the retinal ganglion cells in monkeys and with much lower contrast in humans. And this involves sequentially now moving an offset aperture around across the retinal conjugate focal plane to get access to this multiply scattered light that contains information about those weakly scattering structures such as the retinal ganglion cells. And we had a great opportunity there to validate this with 2-photon imaging in monkey with Robin Sharma and others at Rochester. Dan Ferguson presented at the ARVO meeting in 2018 the first multi-fiber simultaneous offset aperture AOSL imaging of the inner retina. This is some images that are associated with that abstract that is published online. This was a really cool development that I really like and I want to highlight as well is the work by Caitlin Spoznick and Steve Burns' lab. Showing how you could actually make a rapidly configurable aperture with a DMD. And so they were able to use sort of the on and off state of a little digital micromirror device to build all kinds of annular apertures of sort of any configuration you want. So this was a really cool, I think, approach to this type of imaging. And achieve some really nice images. I think these are quite impressive, and I think that at least from my perspective, it looks like there's a lot of signals in these images from the retinal ganglion cells as well. Mozafari et al. In Austin Rorda's lab demonstrated a really nice approach for multi-offset imaging, being able to do it simultaneously now with a detection with a fiber, similar to what Dan had showed at ARVO, and they published this paper showing how you could use an off-the-shelf fiber and sort of configure it with some zoom optics to be able to either do pixel reassignment, which is a super resolution technique, and also offset aperture imaging, and this required an optical mask to sort of make the fiber have sort of a confocal detection at the center, while it had this larger apertures for offset imaging. Finally, the last and most recent one I'll talk about, which is from last year, published by Alf Duber's group, was demonstration of what they call non-confocal quadrant detection. And now adding some more telescopes in here and some more detectors. Alf showed how you could split the light now to 4 detectors and evaluated various configurations of non-confocal quadrant detection. And I'll show some images later from a recent paper that's really highly relevant. Okay, so that brings us up to sort of 2022, and Elena presented some of her work on Wednesday, so I'm not going to go into this in much detail, just to say that her work really was important for us to understand empirically how to configure our detectors so that we could balance this this trade-off between sensitivity and resolution. So we have, opportunity to get more light the closer we go to the PSF center, but also we begin to bring in more of the confocal signal. And so there's sort of a distance at which you can avoid that more directionally backscattered confocal light. But if you go too far, you're really out in a space where there's very little signal. And so in this work demonstrated on the right here, you can see how the contrast is improved as you can improve your detection by including more of the apertures. This was sort of the final configuration that provided the best contrast. Down at the bottom here, J and I, you can see in particular here where using only 2 of the apertures, you can only see some of the structure around what we think here are microglia, but by including all of the offsets, you can start to see the processes of those cells, and you can also see the structures in the retinal ganglion cell mosaic coming up into higher contrast as well. And as others have shown, and we've shown previously as well, this angular dependence that I'll touch on again in just a minute. So Elena showed that this was highly reproducible, at least the structure that we could get from the retinal ganglion cell mosaic. But during this work as well, and doing this repeatability analysis, she observed these cells that appear to move across the surface of the retina. And so I'll be talking more about the work following up on that in just a second. Before I get to that, though, I want to talk a little bit about Pedro Mesia's work with us to extend the Guevara-Torres wave optics model to 2 dimensions. And so this was presented earlier. This is basically just a very simple model showing each cell as a little refractive element within the retina, and as light passes through and experiences a refractive index change, that light will be deviated, allowing that light to be detected then in these off-axis modes. What Pedro showed empirically from our data is that one of the axes actually provides this asymmetric light distribution that gives us high phase contrast, but the perpendicular axis actually provides a symmetric one with substantially lower contrast. And you can see that here in the power spectrum of the images on the right. That lower left of this little animation here, you can see these asymmetries showing up in the power spectrum. Pedro showed that you can actually use the image processing routine to spatially filter the data in frequency space and then fuse the data from the different apertures, only using the information that's maximal about the cells. And this is just a little animation here showing the difference between the conventional combination and the spatial frequency based image reconstruction method. Okay, so what I want to focus on for the rest of the talk is how we moved from sequential to simultaneous detection using a custom fiber bundle, similar to what Dan and Mozaffari et al. Had showed previously. To do this, we devised a custom fiber bundle. This was driven from the data that Elena had collected. And so we settled on this pattern here with 6 apertures. We had been doing 8 in a lot of the previous sequential work, but we moved to 6 here due to some constraints around the manufacturing limitations and practical purposes. So for doing simultaneous detection, we also need an 8-channel or 7-channel really DAC system, and that was provided through our collaboration with Pavan Turuvidula from Austin Rorda's lab, and so he was able to help us get up to 8 channels to be able to do this work. And it simplifies the optical system because we get rid of this telescope, we get rid of this reflective pinhole mask, and we just put our fiber there. And so we have a confocal aperture at the center, and then these offset apertures around it. And the results in healthy eyes showed that the fiber bundle detection has equivalent or slightly better image quality. We could see that there was a little bit less noise. There was a little bit more energy at the bands corresponding to where we expect to see the cells in these images, but it's modest. So this shorter acquisition time allowed us to image more retinal areas and allowed these larger montages to be generated, so we could begin to quantify the morphology of the microglia cells, these cells along the bottom here. We think that these are microglia, and they exhibit a range of morphologies consistent with what we would expect for microglia. Using the fiber bundle offset fibers, we can create perfusion maps. Now, each fiber has sensitivity to different angles of vessels, so you have to sort of fuse all the data from the motion contrast perfusion maps to see the perfused vessel map. But we did that to ensure that what we were counting and detecting were not diving vessels or anything like that. So we or I should say, Yuha Ray, the very talented student in my lab that worked on this project and others working on the project, manually segmented these cells from the normal observers. We detected around 240 cells. The average diameter was around 13 microns. And they all didn't look very, elongated. There's sort of a range of morphometries, including more circular cells, and this is sort of a distribution of this circularity index metric here, eccentricity. But this is the really cool part of what this enabled for us, at least, when we were doing sequential imaging in the work that I showed earlier from Elena's work. It was really hard for us to be able to acquire the images in any short amount of time. So it took several minutes to sequentially move that aperture physically around and get all the light detected. But with the fiber, we could do this and begin to monitor changes over relatively short time scales. And so here we're imaging, the image on the left is every 30 seconds, and the one on the right as well as every 30 seconds. You can see that for the most part, these microglia normalized don't move very much. One of the interesting things that you can see is we can see in some cases the processes of these cells extending and retracting. In the center here, this is over the course of every 5 minutes, every 30 seconds. And the right here, you can see with the images or the cells that are called out with the arrows in the image, that for the most part, these cells are mostly stationary and normalized. So we did the quantification and showed that these cells are really not moving that quickly and most normalized. The most motion that we had detected and normalized is this video on the left. And one of the cell tracks here is that the fastest cell in that video, which was the fastest one we detected. We could also image areas that were more difficult to image, especially with our sequential approach. Those of you that do AOS low, you know that imaging this area near the optic nerve head is particularly challenging. And we detected some cells in here that we're not sure what they are. They're much larger than the microglia. They're about 25 microns in diameter. So if anyone knows what those cells are, please reach out to me. I'm trying to figure it out. If you have any histology, that would be helpful. So finally here, getting to imaging of patients, we brought in some patients from the uveitis clinic, and one of them that we had the opportunity to look at was a patient with acute syphilitic posterior placoid chorioretinitis. So these patients have syphilis bacterium that invade the eye, and it's a really really bad infection, and we could see a lot of activity in these eyes. We could see morphologies that look like macrophages. We could track these cells over time. We could see some that move very rapidly, like these ones moving across the screen right now. And we quantified their motion and compared with normal eyes. It's really best, I think, to look at this stuff in motion. So this is 3 imaging time points now, the second one 3 weeks later. And the third one at day 35, and we can see now how the number of cells is reduced and their motility is reduced. And by the final time point, we even begin to see some cells go back into that ramified or resting state. All right, so I need to speed it up here because I'm running out of time, but I sort of already said that their speed and motility decreased with time. This is just another example that we have of patients with uveitis that I want to show. On the top here, this is a person that has a chronic uveitis that we think is well controlled on immunomodulatory treatment. So we don't see a lot of cell activity there, whereas on the bottom, we see one with an active infection where there's all kinds of activity. This is very strikingly similar, at least to me, to what was shown by Jesse Shallak and his group. A.B. Joseph from his lab had this really nice paper in 2020 where they induced a really nasty acute inflammatory response in these mice and could monitor these cells over time. Of course, with the mouse NA, you get much better resolution. I just point out, I think we've seen these cells in some of our previous studies, and I'm not sure if these are the same cells that were imaged by Migaza et al. That was published just this year. These, they claim, are the vitreous cortex hyalocytes, and they provide really good data to suggest that. However, there's no RGC signals visible in their images, and they do this embossed filtering approach that gives them high contrast to the processes, but really obliterates the background and gives this pattern that they claim is the web-like pattern is the vitreous cortex. All right, I'm just going to wrap it up here and just show one last piece that these cells have been imaged by AOCT as well. And so we need to figure out if these are the same cells and why they're not seeing them in the ganglion cell layer, because that's sort of where we think we are. So just to conclude here, this simultaneous detection really simplifies the optical design, improves the efficiency for multi-offset. It allows us to see microglia and novel cells in the retina and study their morphology and dynamics. We think this is going to be really impactful for looking at other conditions as well, like inherited retinal degenerations and AMD. And I just want to emphasize this can be added to any existing AOS lows with just some modifications to be able to detect additional channels. So we can't distinguish, what can't we do? We can't distinguish the macrophages from monocytes that infiltrate the retina. We really need to improve the segmentation because doing this manually is too tedious. And we need multimodal comparisons with AOCT and histology to really understand what these cells are and these subcellular structures. So we need to do more image processing and more work. And so, sorry I took a couple minutes too long there, but thank you for the opportunity here to present. I just want to acknowledge my really great team. And if there's any time for questions, I'd be happy to take any. Thank you.
Thank you, Ethan. I think maybe we try one question. Please. Well, you can try to say it and then I'll repeat it.
I was wondering, are we able to distinguish between ramified microglia and say like amoeboid, so like active microglia, as to whether they're actually imposing an inflammatory threat or if they're just surveilling? And if you were able to distinguish any differences between, let's say, inflammatory conditions and
so did you hear part of that though?
A little bit. Something
Yeah.
ramified from activated microglia. Is that the question?
Yes, yes. Whether you can see any differences between them.
Yeah, so we see some hints that we can begin to see different morphologies over time exhibited. And there's this case here where we see these very amoeboid-like structures at the first time point, some of which look like they have internal granularity that we think are macrophages. So that they could have that sort of macrophage-like activated morphology. And then later in time, there's at least a couple cases, they're sort of faint, and I don't know how well they're coming across over the Zoom, but there's in day 35 for the bottom middle of this image, we can see a ramified now microglia with their processes extended. But I think one of the things that we might really need to do is improve the image processing to be able to better detect the processes of the cells so that we can distinguish the activated from the ramified. But that's a great question.
Thank you so much, Ethan. Thank you.
Thank you.
So the last speaker of this session is Alexander, please.
This one.
So we might have to move things around a little bit. Let's see.
Where? On the right.
Yeah, but you can also move the.
It's okay.
That's what. Please.
Okay, thank you. So I will talk about adaptive optics in non-human primates. Just to say, compared to the previous presentation, which was very technical, I will be very basic on this presentation. So I will talk about flood illumination, adaptive optic flood illumination ophthalmoscopy in non-human primates. So you may ask, why is it interesting? Because obviously it's been a decade that we're using the RTX1 in humans, in clinical studies, in preclinical studies. So I will just talk to you a little about the background of why is it in my interest to present you that. So in our team, in the Institute of Vision, we basically want to aim to create retinal degeneration models, and we want to apply it on non-human primates, especially on macaques. So I was looking in the literature if there was any description of adaptive optics flood illumination ophthalmoscopy, especially by the RTX1, because we get one in the facility. And in fact, I just found one paper talking about the. Yes, sorry.
Let's have a little break. It seems like it's not shared. Is that the case?
Okay.
Can you not see his presentation?
Yeah, we can't see the slides.
Okay, sorry. So you have to move the Zoom screen back. Shall I do it?
The green. Can I?
The green on the
Excuse me. Yes. Which one? One or two?
It's this one.
But better take this one, because when you pull full screen,
And
there we are.
present.
And then, oh yeah. Okay.
On the top, maybe.
This one.
Yeah.
Is it okay?
How does it look? Can you see?
Is it okay?
Yes, no, it works.
It works? Okay.
Okay. Thank you. Sorry for that. So I was saying that I found only one study that was talking about adaptive optics using the RTX1, sorry. And it was on Japanese macaques on a model of AMD. In fact, these macaques are developing a lot of drusen. So they didn't really talk about normal retinal features. And so I was thinking, okay, in non-human primates, there is quite nothing, and in non-human animals. So I found one study from Serge Rosolant, who reported adaptive optics by flood lumination only in cats. So because this picture is heartbreaking me, I wanted to put a happy cat on it because it's
after the experience.
Yes, of course. Okay, so the goal of this is just to show you that between humans and non-human primates, it's practically the same. So I can see here, and on the Zoom, there are many of, let's say, experts in this area. I wanted to challenge you a little bit on this. Here is one picture. One side is a 5-year-old male. It's a cynomolgus macaque, Macaca fascicularis. And on the other side, there's a human, 20-year-old male, and who was healthy. So what do you think? It's not obvious, isn't it? So here you are at 2 degrees of eccentricity at the temporal border of the foveal pit. The fovea is marked into the dashed circle. So if you look closer on picture you are more familiar with, the cone mosaic appears really the same. You can maybe say that on the left side, the cone mosaic is more homogeneous, let's say, but it also depends on the acquisition. So, and in both cases, you have brighter and darker cones that have been well described with the RTX1 in humans. So, in fact, it's the left one is the primates and the right one is the human. Okay, so that's not easy to say. So, just to show you a few features of adaptive optics in non-human primates, I just wanted to focus on the F panel, just to show you that on the left side of the F panel, you have an adaptive optics imaging with the RTX1. And on the left side, on the right side, sorry, it's immunolabeling with the peanut agglutinin. So it's labeling cones in the non-human primate retina, just to show you that the aspect is the same. And you know, when you're imaging with the RTX1, you can have those cones with, let's say, a bright shadow just beside it, and it can expect it to be like the outer segments, which is a little bit, you know, deformed, and you can see it as well. So we wanted to make the parallel between those images. It's not the topic here, but it's just to say that the photoreceptor layer look the same, but it's also the case with the vasculature. And later I will just talk to you about induced retinal detachment, because it's a way that we want to induce the degeneration in primates. And this is just to show you the injection site, which is at the branch of the upper temporal vessels, obviously in the right eye here. This was also to show you quantitative analysis to ensure that the embedded software, so how it detects, as you know it well, works in non-human primates. So you've got humans on the top, non-human primates on the bottom. The semi-automated detection of cones works really well. And here you're at 2 degrees of eccentricity, and you have a cone density that it's really the same between the primates and the human. Here, cone density is in cones per square degree, because, as you know it, the actual length between human and non-human primates are not the same. In human, you're about 23 millimeters. in non-human primates, it's more about 17, 18 millimeters. Another way to quantify the cone mosaic is to do the power spectrum. Here is it on the right side. And you can see that the power spectrum is decreasing, let's say, with the eccentricity as well in the two species. Here you have the quantitative analysis of cone density in B and C, which shows a decrease in density with the horizontal and vertical eccentricity. The dashed line represents mean values of 4 healthy humans, and you have 4 non-human primates in color. Also, we found a strong correlation between cone density, cone regularity, and cone spacing values between the two species, with a Pearson R, which is around 0.9. That image just to show you that it's not quite easy to get these measures on primates, because, as you know it, the RTX1 is not, it's not like the spectralis. Let's say you can't move it in the 3 dimensions. So you need the primate to be moved in the 3 dimensions. So it can be a little bit hard, but if I figured it out, you could do it as well. So we also wanted to investigate the effects of an induced retinal detachment using DMSO, because it's the vehicle of our toxic agents that we use to induce retinal degeneration. So we delivered DMSO in the subretinal space, and we wanted to detach the superior fovea. So here are the multimodal imaging using the spectralis, including the OCT. You can see that the induced detachment, so detached the superior fovea, and the retina completely reattached at day 3. Maybe you can see here, but I don't know if... No, you can't see my mouse, but at day 3, the interdigitating zone on the OCT is a little bit, has a little bit a ratty appearance, let's say. So you may conclude that photoreceptors are not normal, but we wanted to see with adaptive optics. So we managed to get at let's say a borderline of the detached area and the undetached area. So the detached area here is represented, the power spectrum of the detached area is represented in the gray line, and the black curve is the detached area. So you can see that here we are at 15 days after the induced retinal detachment. So the retina is reattached. And you can see that the power spectrum plots is like there is no cone peak. Here is the evolution during 4 months after in the same region of interest. It's at 2 degrees of eccentricity. You can see the baseline at the top. Then 3 days after the detachment, 15 days, 1 month. And at 4 months, the power spectrum kind of returned to normal. So that was interesting because it's in accordance with studies that were published in the 80s and the 90s that studied induced retinal detachment in primates and showed that the outer segment length recovered fully 4 to 5 months after the induced detachment. It's just for your information, it's not quite the topic here, but we also did functional testing of the retina using full field electroretinogram and multifocal electroretinogram, and it showed no function alteration of the photoreceptor in the detached area. So it seems that in adaptive optics we can see alteration of the structure, but there is no alteration of the function. So that's quite interesting, and we just proposed based on this study, on these results, and on previously reported studies on various species, we propose scheme, let's say, of the photoreceptor and the RPE interaction when you're inducing a retinal detachment. So when the detached retina, you have no contact between the outer segments and the apical membrane of the RPE. And when the retina is reattached, let's say in the early reattached retina at day 3, for example, you have interactions beginning. Then it's well described in the literature that the outer segments are being less, the outer segments, the length of the outer segments is reducing, and then you get to approximately a normal aspect at 4 or 5 months. Thank you. I want to just do some acknowledgements. So to the Paris eye imaging team that helped me a lot for quantifications, to Marine Durand, who helped me a lot to set up the RTX1 at the NHP facility. And I wanted to thank Région Île-de-France, the Foresight Institute, the Fondation Voir et Entendre, and the Fondation pour la Recherche Médicale, who funded this project. Thank you.
Thank you so much, Alexandre. Any questions? Oh, several online. Or maybe it's because, yeah, let's see. Can an admin please mute? Oh, okay, we didn't get that. Yeah. Sorry. Any questions? Yes, Kate, please.
So are the ERG results sensitive enough to pick up small scale changes? Do you think you could use another method?
That's very interesting. In fact, as I said, so we also delivered a toxic molecule in the subretinal space, and we of a non-human primate, another one, and we also analyzed with multifocal electroactinogram, and it was it showed differences, let's say, between the eyes injected with the toxic molecule and the eyes injected with just DMSO. So let's say it's sensitive enough to show an alteration of the function, but I don't know, maybe an hypothesis could be that it has been shown that after retinal detachment, there is kind of an obscene redistribution in the inner segments. So maybe it can explain that there is an alteration of the structure without a functional alteration. But yeah.
Thank you.
Thank you.
That was the last of that session. So thank you to all speakers and for everyone to sit and listen to us. Thank you. I don't know how you want to do with the break.
So we'll take the break until around 4:35. So
4:35. So we'll be back at 4:35. Thank you.